The overall goal of this procedure is to visualize living neutrophil granulocytes and extracellular DNA nets in a lung that has been recently infected with a fungal pathogen. First, mice that are positive for EGFP in their neutrophil population are infected with conidia that are also fluorescently labeled. Next, the infection is allowed to develop over 7 hours, after which the infected animal is sacrificed. Then, the lungs of the infected animal are filled with low-melting agarose to facilitate sectioning by vibratome. Finally, a thick slice of the infected lung tissue is acquired via vibratome, and lung neutrophils, DNA nets, and fungal elements are imaged. Ultimately, high-resolution still images or movie sequences of granulocytes in live lung tissue can be obtained through multicolor two-photon microscopy.
The main advantage of this method over existing technologies is that the immune cells responsible for clearing the infection can be observed directly within the natural environment. This can be done during the process of net formation and fungal killing, and thus allows a much less artifactual analysis of neutrophil physiology.
I invented this technique when Matthias annoyed me with his burning questions regarding the in vivo relevance of my prior in vitro findings, and I will present this technique in the next couple of minutes.
To generate swollen conidia of the environmental mold Aspergillus fumigatus, add 5 ml of medium to a 6-well cell culture plate. Then, transfer 2 × 10⁷ resting conidia to the plate and incubate the spores for 7 hours at 37 degrees Celsius in 5% carbon dioxide in a BSL-2 facility. After the swelling period, fluorescently label the spores by adding 10 microliters of calcifluor white stock solution to each well. Following gentle mixing, incubate the plate for an additional 15 minutes under the same conditions. To harvest the fluorescently labeled spores, place a 70 micrometer cell strainer on top of a 50 milliliter conical tube. Collect the spores from the plate by pipette and then pass the media solution through the cell strainer into the tube. Next, spin down the cells at 900 Gs for 5 minutes at room temperature and discard the media. Then, wash the spores with 10 milliliters of PBS under the same centrifuge conditions. Resuspend the spore pellet in 2 milliliters of PBS and determine the total number of swollen conidia by cell counter. Spin down the spore suspension again under the same centrifuge conditions as before, and afterward carefully remove the supernatant. Resuspend the sedimented conidia in a final concentration of 1 × 10⁷ cells per 100 microliters of PBS. To prepare the mouse for infection with the spore solution, the animal is anesthetized and sedation is confirmed by lack of response to toe pinch. In this video, lice EGFP mice, which carry EGFP under the lysozyme promoter and therefore have EGFP-positive neutrophils, are used. 5 minutes after administration of anesthesia and confirmation of sedation, use an elastic band to suspend the narcotized mouse on a bevel by its teeth. Under a gooseneck lamp, use forceps to pull the tongue to one side and insert a 22-gauge indwelling venous catheter into the animal's trachea. After successful insertion of this tubing, use a 100 microliter micropipette to administer 100 microliters of the spore suspension. Then, to enhance the distribution of the fungal particles throughout the lung, mechanically ventilate the infected animal for 2 minutes with a small animal respirator at a breathing rate of 250 breaths per minute and an inhalation volume of 300 microliters per breath. Finally, remove the catheter and place the mouse back in its cage for 7 hours while the infection progresses. Observe the recovery of the animal every 5 minutes until ambulatory again. Just before the end of the infection period, melt 20 milliliters of low-melting agarose in a microwave and keep it warm so that the agarose does not solidify before it is injected. Chill a bottle of PBS to 4 degrees Celsius in the refrigerator at this time as well. At the very end of the infection period, the animal is euthanized. Use needles to fix the mouse to a silicone preparation mat and carefully open the chest using a pair of scissors and forceps to expose the lungs and upper respiratory tract. Starting from the exposed epiglottis, insert another 22-gauge indwelling venous catheter into the trachea. Using a sewing needle, a standard sewing thread is layered around the trachea as preparation for the subsequent agarose filling step of the lung. Next, fill a 1 milliliter syringe with the pre-warmed agarose and administer the solution through the catheter to fill the lungs with it. As soon as the lungs are filled, tie the trachea closed with the prepared sewing thread while removing the catheter, and put the whole animal into a refrigerator at 4 degrees Celsius for 15 minutes. After the agarose has solidified, beginning from the trachea, excise the whole lung from the opened chest and wash it with the pre-cooled PBS. Using a pair of sharp pointed scissors, remove the right lung lobe. Briefly dry the bottom of the lobe on a sheet of tissue paper, and then fix the lobe to a vibratome preparation block using a drop of tissue adhesive for 1 minute. Next, install the block in the cutting chamber of the vibratome and then fill the chamber with the pre-chilled PBS. Set the razor to a mid-range vibration with a medial driving setting and cut a horizontal cross-section of the lung. Then place the upper half of the sliced lung tissue upside down in a 5 cm diameter plastic petri dish. Fix the lung tissue to the petri dish by placing a flat washer on top of it. Fill the dish with 10 milliliters of PBS and add 10 microliters of Cytox orange dye. Finally, install the petri dish containing the lung tissue under the microscope objective so that the buffer can be warmed by a temperature sensor-controlled heater to 37 degrees Celsius. For imaging, different areas along the dissection are scanned down to 400 micrometer depth using an illumination wavelength of 800 nanometers, detecting green, 530 nanometers, and red, 580 nanometers fluorescence, as well as the second harmonic generation, or SHG signal, and the blue calcifluor fluorescence at 400 to 470 nanometers emission with external non-descan detectors, or NDD. Shown here in blue is the SHG signal of the alveolar tissue structure of a non-infected lung of a C57 Black 6 mouse. Note the fibrous tissue at the bottom of the lung slice as compared to the clearly alveolar organization within the breathing active areas at the top of the lung. In this figure, the Cytox signal of lung cell nuclei cut open during the preparation of the lung slice are visualized in red. This figure shows an overlay of the two channels, with a close-up of the boxed area of the tissue shown here. Here, the blue SHG signal indicates the alveolar and fungal structures present in a lung slice of a C57 Black 6 mouse infected 7 hours prior with A. fumigatus. In this figure, the red Cytox signal shows not only the nuclei of lung cells cut open during the preparation of the lung slice, but also the DNA nets. Here, an overlay of the two channels can be seen. In this enlarged graphic of the boxed area from the previous figure, distinct areas of fungal masses, alveoli, and net structures are indicated with the letters F, A, and N respectively. The combined fungal structure and SHG of the alveolar tissue, blue, the Cytox signal of cell nuclei and nets, red, as well as numerous lung-infiltrating neutrophils, green, can be visualized here in a slice of lung tissue from a lice EGFP mouse 7 hours after infection with A. fumigatus. Neutrophils, in green, can be seen migrating and phagocytosing fungal elements, in blue, in Aspergillus-infected lungs, as seen by time-lapse two-photon microscopy of a living lung slice 7 hours after infection. Cell nuclei and net structures are depicted in red. The real time of the experiment is shown on the lower right, and the scale bar is indicative of 50 micrometers.
Though this method can provide insight into fungal infections, it might also be applied to other systems like viral or bacterial infections, and it might also give insight into the function of other resident immune cells inside the lung.